Now. Okay. So welcome. Now it's sharp 4 o'clock. So I will introduce our first speaker. So it is Léa Hiéronimus of the UCLouvain, who will present his work on a 3D model to a simplified 2D macrophage bioassay predicting fibrotic activities of inhaled particles. So he obtained his master's degree in Biomedical Sciences at the UCLouvain and is now doing his PhD in the Louvain Center for Toxicology and Pharmacology. And the subject of his thesis is on the hazardous effects of inhaled inorganic pollutants, which then specifically on the formation on lung granulomatous fibrosis. And he is developing new simple bioassays relying on cell lines to help industry predict materials that will induce this pathology. Léa, the floor is yours.
Thank you for this introduction. I'm sharing my screen. And yes, I think you can see it. So as you said, I'm in the middle of my PhD thesis, and I study granuloma, or particle-induced granuloma, which is what you can see here in this biopsy. These cell nodules, which are formed in the lung after inhalation of inhaled inorganic particles, such as silica, which is the case of this biopsy. It was a 70-year-old worker in the clothing industry sending denims. The inhalation of a large amount of silica particles led to the formation of these nodules called granuloma in the lungs. The goal of my PhD thesis is to better understand the mechanism allowing granuloma formation, how to better treat this pathology, and to help prevent it. I'm going to show you. So a granuloma is by definition a compact aggregate of macrophages or other cells surrounding an agent, which can be a microorganism or an antigen recognized as foreign to the organism. So it can be a chemical, it can be suture, piercing, but in case of lung granulomatous disease, it's often inorganic particles. It can be caused because of infections, such as tuberculosis. Certain drugs can induce lung granulomatous diseases. Those are, for example, against a certain cancer. And they can also have an unknown cause or linked to genetic predisposition or with an autoimmune component, which could explain how certain autoantigens are now recognized as being foreign. These are lung granulomatous diseases. Yeah. The consequences of this pathology is decreased lung function, as these granuloma take some room and decrease the surface available for gas exchanges. Granuloma also increase the risk of developing additional pathologies, such as lung fibrosis, lung infections, autoimmune diseases, and cancer. And actually, I think it is up to 20% of idiopathic pulmonary fibrosis patients which have been exposed to inhaled inorganic particles. In terms of treatment, there is no cure. Once the granulomas are there, we can only wait for their resorption. There's only close follow-up and symptoms management and treating the triggers if it has been identified. So this is the clinical point of view, but in terms of biology, what happens? Okay, so in particle-induced granuloma, here I'm going to explain what occurs in what has occurred in the lung of this patient. The foreign body is a pollutant, an inorganic micro-sized pollutant. For example, it can be silica particles. Once it enters the deep lung, it will be recognized by immune cells, which will try to remove them. They often do so through phagocytosis. This is how macrophages will try to clear the lungs. They will eat the particles. Sometimes it works, and it allows the removal of the particles. But if the particles are biopersistent, macrophages will become activated. Activated macrophages will release cytokines and chemokines, which will activate other cells, macrophages and new immune cells, which, because of this activation, will try to follow the gradient of chemokine, leading to an aggregation of cells surrounding the cytotoxic particles. These cells will become activated, release more and more chemokines, leading to a cellular aggregation surrounding the particles. In granuloma formation, there are three main mediators known in the literature. Two of them are CCL2 and osteopontin. There are important chemokines also acting as cytokines. Their levels correlate with the number of aggregated cells in many models, and rodent knockout models show that the absence of CCL2 and osteopontin reduce granuloma formation and granuloma fibrosis. And it is also the case for models where animals have been orally treated with certain inorganic particles called carbon nanotubes. So these inflammatory cocktail will lead to the attraction and activation of other cells, other macrophages, but also lymphocytes and neutrophils. All of these will try to remove the particles. Sometimes it works, sometimes the particles are biopersistent, and this inflammatory cocktail will slowly change and lead to a fibrotic granuloma. At this stage, new cells are added, epithelial cells and fibroblasts will invade the granuloma, further isolating the cytotoxic particles from the healthy surrounding tissues. At this stage, CCL2 and osteopontin are still important, but there are new one, and the third one is TIMP1. TIMP1 is produced by fibroblasts, but also epithelial cells and macrophages. In healthy conditions, TIMP1 allows the proliferation of fibroblasts and their synthesis of collagen, constituent of the extracellular matrix. In a healthy condition, new collagen fibers are formed and old ones are removed thanks to collagenase. This is the healthy, balanced turnover. But when there is an excess of TIMP1, which is the case in fibrotic granuloma, the excess of TIMP1 leads to collagen accumulation, and TIMP1 also acts as an enzyme, as an inhibitor, as TIMP1 is tissue inhibitor of metalloproteinase 1. It inhibits collagenase, which amplify the accumulation of collagen. What does it do? Well, this collagen leads to a fibrotic core with necrosis, and the collagen fibers will accumulate in the lung surrounding the granuloma. It leads to fibrotic granuloma. The collagen fibers rigidify the lung, further decreasing the lung function. So this is the chronic stage of the granuloma. Now that I've explained the cells important in and the three mediators important in granuloma formation, the goal now is to explain you the why does granuloma occur. In the case of this 70-year-old worker, he has inhaled many crystalline silica particles. Industries are creating new and new materials, and sometimes the materials can leak certain particles. Among these particles, certain are inducing granulomatous disease, but others are safe from inducing this pathology. We know, thanks to historical use and animal models, that carbon black and tungsten carbide will not induce this pathology, while asbestos, certain silica particles will induce granulomatous disease in humans. And for carbon nanotubes, as I've said, there are animal models, but humans are not exposed yet to carbon nanotubes. So to help industries better identify which of these particles will induce the pathology. Currently, the only acceptable method is using in vivo models, but thanks to 3R legislation, things are changing and new models are being developed. And I'm going to present you the model on the Replace platform that I've created in this context. Okay, so what I've done is creating a particle-induced granuloma model using cell lines. I've used 3 different mice lung cell lines, MHS macrophages, and fibroblast and epithelial cells cell lines. First, macrophages are pre-exposed overnight to different particles, inducing or not granuloma. After overnight exposition, they are collected and co-cultured with fibroblasts and epithelial cells to mimic what occurs in vivo. After a week, they are co-cultured in 96 well plates coated with scaffold, either agarose gel or matrigel, and the cells are followed. I followed the cells for a week using an optical microscope. As you can see here, the cells are not specifically organized, or they are in a monolayer in the vehicle condition. When macrophages have been exposed overnight to carbon black, which is not granuloma inducing, there's no clear difference, or it's hard to tell. When macrophages have been exposed to granuloma inducing carbon nanotubes, the cells are more organized, the aggregation is more compact. Maybe there are some layers, but I agree it's difficult to tell. So after that, I've repeated the experiment and used different cell staining. Before the co-cultured, I've stained macrophages in green, fibroblasts in blue, and epithelial cells using red cell dye. Okay. And I've used this time confocal microscopy. In not granuloma-inducing condition, the cells are not organized in any specific way, but in granuloma-inducing conditions, we can see spheroids, which are organized in layers with a fibrotic core and with fibroblasts and epithelial cells, and a corona of macrophages in green, having phagocytosed the carbon nanotubes in black, but you cannot see the black. So after a week, you can see this clear spheroid formation, which has been induced by granuloma-inducing particles. The spheroids, I went through the cells, through the different layers using the microscope to show you the 3-dimensional organization of the cells. In vehicle condition, there's no specific organization, neither for carbon black, but in granuloma-inducing conditions, you can still see the organization with this round, compact fibrotic core, and macrophages in green surrounding this spheroid. For sure, the macrophages in green are kind of shy in this condition, as you cannot clearly see this big corona of macrophages, which was in the previous slide. It was another day, another experiment, and here I think macrophages after a week have been more difficult to maintain in culture, but they still allow this formation of this organization that you can see. So the formation of this granuloma was progressive. You cannot see it in not granuloma-inducing conditions, but carbon nanotubes induced this progressive formation of a granuloma, culminating in after a week. This is qualitative data. You can see and judge the formation of spheroids or not. It's good, but we still would wanted to have quantitative data. So what I've done is collecting the supernatant of these wells and measured using a simple ELISA technique the mediators that we know are associated with granuloma formation. I've measured CCL2 and osteopontin, and there's no difference in the levels of these mediators. Why? I don't know. There were granuloma formation in there were granuloma in the wells, but these mediators were not significantly elevated in the CNT conditions. I've measured TIMP1, which is significantly elevated in the wells containing the spheroids. However, MMP1, which is a target, one of the targets of TIMP1, is not modified. This shows the unbalanced in extracellular matrix remodeling, which is favorizing TIMP1 and the switch from the inflammatory granuloma to the chronic fibrotic one. I've repeated the experiment and collected supernatants from different days after coculture. And here again, you can see the progressive, significant increase of TIMP1, specifically in granuloma-inducing conditions. So this shows qualitative and quantitative data using this model using 3 different cell lines. But 3 different cell lines and using confocal microscopy may be too much for industries, as maybe they would have liked some simplified model. What I've done next is to try to remove fibroblasts and epithelial cells from my model. Because in this case, only macrophages were directly responding to particles. So I've tested this hypothesis and removed fibroblasts and epithelial cells from my model. And as you can see, macrophages are sufficient for the formation of spheroids induced by granuloma-inducing particles. These spheroids are not organized in layers. They are not as well organized as they were in the 3 cell lines model, but you can see granuloma formed only in response to particles that are known to be granuloma-inducing. I've also measured the levels of TIMP1, and macrophages alone are sufficient for the release of TIMP1 in granuloma-inducing conditions. So this is a simplified version of the model, which I've been using for my PhD thesis. My PhD thesis did not focus on forming a new granuloma model. I wanted to do this model to study my favorite cell type, which are B1 lymphocytes. These are one of the lymphocytes that are recruited from the peritoneum to the lungs during granuloma formation. And in the lab, we have accumulated ample evidence showing that these cells amplify granuloma formation. So I've used my model to study how these cells amplify granuloma formation. I've added B1 cells to my model and found that granulomas were larger and more organized when adding B1 cells, and the spheroids will form only in granuloma-inducing conditions. And also, B1 cells stimulate macrophages into the release of more TIMP1. As conclusions, I have developed a simple model using only 1 cell line, which yields both quantitative and qualitative results for in under a week, which is faster and cheaper than what is necessary in vivo. It has application for both fundamental research and predictive toxicology. This is why I've added this model on the Replace platform. In terms of perspectives, I'm currently no longer adding particles on a concentration that is based on mass, but on their surface area. I'm planning of using a wide range of particles which are known to induce or not granuloma and or fibrosis, and to replace these MHS macrophages by a human cell line or macrophages purified from human lungs. I would like to thank my supervisor, François Yo, and my colleagues in the lab. And you can find the model under Replace platform under the same name. Thank you for listening to me.
Thank you very much, Léa, for your nice and clear presentation. You have already done a lot of work in the middle of your PhD. It's also nice to see that you have incorporated then 3 different cell types in your model and that you use it to simplify it for industry, and then more in particular that you're also planning to use human derived material in the next steps of your project. So that's very nice and also fully compliant with the 3 R's principle and very relevant for human-based technologies, obviously. So I'm looking to Mo to see if we have any questions.
I think in the chat I don't see any questions, but maybe I can ask one myself. I was wondering, so you said that your presentation, your work would be very useful for industry. So I was wondering which type of industry would benefit from this work, and if you plan later on to work towards the regulatory acceptance of your methodology.
In terms of industries, there are some, like I said, silica particles are used in the clothing industry. There are also new materials, which there are some aerosols containing these inorganic particles. But lithium-ion batteries also had a project of doing paint-like batteries, which meant the inhalation of these inorganic particles. In a way, every industry which has the potential inhaled exposure could want to use this model to narrow down the materials that they want to put on the market, which would help them testing on in vivo models only few particles and to remove the ones that are clearly granuloma inducing using this model. In terms of regulatory acceptance, I'm not at this stage yet, but I would hope that someone would want to do this process, but it's a long process to become a validated method. But maybe it could help industries.
Mm-hmm. Yeah. Okay. Thank you. Then I don't see any other question in the chat, so I think maybe we can move on to the second presentation.
So thank you very much, Léa, for your presentation. It was very nice to see the progress of your work. It was a very nice presentation. And then we also hope, obviously, that in the future, your method will be validated and then it can be then routinely used by industry. So that would be very good news, especially because indeed the nanoparticles are becoming more important. So very valuable work, of course. So then indeed we can go to the next speaker, who is Dr. Amar van Laar. So he's from the University of Ghent, and he will discuss his work on the metabolism and health effects of rare sugars in coculture modules. So Amar obtained Biomedical and Nutrition Science degrees in the Netherlands before starting his PhD in Ghent University, and he has already contributed to several molecular research projects on nutrition and health, mainly focusing on metabolism and cancer. And within these projects, he has been working on the development of innovative in vitro models, which are very relevant, of course, for human research and animal welfare perspective. So Amar, the floor is yours.
Thank you. All right. I hope it is visible now. So indeed, my presentation is about co-cultures as an improved method for in vitro simulation, in this case in the context of metabolic health. I'll start with a brief introduction of my former research. Then I'll give a bit of explanation of the basics of cell culture in order to understand the model that we've been using, which I will explain in more detail, the co-culture models. Then shortly discuss the results and how this could be for the replacement of animals. And then finally, conclusion, room for questions, and a list of publications for people who desire to read more. So I've been contributing to a couple of different cellular in vitro research projects on nutrition and health. First in Wageningen, investigating anti-cancer effects of omega-3 metabolites for colon cancer. Then immunological research with primary immune cells related to beta-glucans to enhance cancer immunotherapy. And finally, this presentation will be about my PhD research on the metabolic health effects of rare sugars. No longer affiliated to these universities, as I'm currently looking for a new challenge. Let's first have a look, why are we interested in those co-culture models? So we were investigating the health effects of rare sugars and more specifically rare disaccharides. And here in this picture, we can see that disaccharides consist of two monosaccharides, and they are digested largely at the brush border of the small intestine. Their disaccharides are broken down in those monosaccharides, which are then taken up, end up in the bloodstream, and are available for other organs. So when you want to investigate the health effects of those rare disaccharides on metabolic organs, such as the liver, you will need such a digestion step, and conventional monoculture models cannot allow that. Within a co-culture model, you can combine different organs, so that's why this is so relevant for us. In cell culture, you culture cells in a flask under stable conditions in a CO2 incubator, so temperature, humidity, CO2 concentration is all stable. It contains, you provide the cells with a medium containing nutrients and growth factors, in our case a DMEM. And then when the cells have grown enough, usually about 80% confluency, you can start transferring them to a cell culture plate. Because we use adherent cells, you need a trypsinization step in which the cells leave the bottom and get in a suspension which you can transfer to the plate. You count the number of cells to ensure that every experiment has the same number of cells and also the same number of cells in each well. And then you're ready for exposure. We expose with sugars, which means that we use a seahorse base medium without any glucose or pyruvate in order to really highlight the effects of the actual sugars that we are testing. And then after exposures, you can measure changes in physiology. If you now go to a co-culture model to the right, you can see that the model has two layers. So you have you can add intestinal cells to an insert, that's a top layer, and then liver cells to the bottom of a plate, which is like a normal monoculture plate, but such a co-culture has an additional layer on top. For our co-culture model, we'll go a little bit more detail about our co-culture model. So we were using CACO2 cells and seeded them in a 24-well trans well. So they are in the insert on top. And these cells need to differentiate for 21 days, starting from the moment that this monolayer has formed. This is because CACO2 cells are more like colon cells, but they differentiate spontaneously and then obtain a more enterocyte, so small intestine-like phenotype, during which they obtain the activity for enzymatic digestion of sugars and also for the transporters. We see the HepG2 cells, the liver cells, at the bottom of a plate. but I would advise not to do that in the same plate, but in another plate. Then you can control it better. And when they are both ready, you just take the insert and put it on top of the second plate. We also perform tear measurements, so transepithelial electrical resistance measurements, which is with a probe. And you can do that every day. You can really monitor the intestinal barrier integrity. So when the barrier grows and becomes thicker, you will see an increase of those tear values. In case of a disruption of the layer, which is really problematic because the exposure can just leak through and you're not measuring intestinal absorption anymore, you can also see a steep drop in tear values. So that's also nice to monitor. Well, after that, you can start exposing the cells. And in our case, we were interested with this model to study non-alcoholic fatty liver disease, which is one of the most common metabolic diseases. And for that purpose, we were testing combinations of sugar and fat. We had one setup in which we just tested sugar solution, so adding to the intestinal cells and then measuring effect at the intestinal level and at the liver. We're also testing combination of sugar and fatty acids, in our case a mixture of oleic and palmitic acid. And then finally a setup in which the sugars are added to the intestinal cells and the fatty acids directly to the liver. We measured different parameters, so tear as discussed for intestinal layer integrity, also lucifer yellow. We perform that only once, so don't monitor that, but that's just at the end. And that's yellow dye that would leak through disrupted barriers. Also performed RNA sequencing to measure gene expression, rhesus-rin measurements for the energy metabolism and toxicity effects, and AlipoRet to measure the intracellular fat accumulation in the liver cells after these exposures. For the next model, we go even one step further because we also wanted to know the effects on insulin sensitivity and insulin resistance, which we measured in skeletal muscle cells. So we went for a triple co-culture model, which now contains 3 different layers. So the KCO2 cells are now added in a 12-well transwell insert. That's again the top layer. These wells are larger than the 24 well, so you have to increase the number of cells proportionally with the surface area of the well. Now the liver cells are also added to an insert, to a transwell insert, a 6-well transwell insert that will now be a middle layer, and skeletal muscle cells at the bottom of a plate, at the bottom of a 6-well plate. And this setup is a little bit more complicated because the 24-well transwell with 2 layers is something that you can buy commercially and it just works. In this case, you need some kind of supports to keep those layers apart. So we have metal supports in our lab that suit the 6-well format, and you can keep those layers apart by putting the supports between each layer. If you don't have those, you might have to do 3D printing or engineer that in order to fit exactly your desired format. So for this, we also measured glucose uptake, because glucose uptake decreases during insulin resistance in skeletal muscle cells. We performed Western blot and also sugar digestion was measured. So the concentration of those sugars was measured over time to study the digestion. Well, by measuring all these different things, we found that the model was able to provide some interesting results. So first of all, with this model, we were able to identify that rare sugars are more slowly digestible than conventional sugars such as maltose as a breakdown product of starch, with also differences between different rare sugars. We see that, for example, Cojibios nigros are still relatively quickly digestible as rare sugars. But in addition to that, we were able to find effects on the energy metabolism, not only in the intestinal cells, but also in the liver cells that are co-cultured with them and are more indirectly exposed to these sugars. Same is true for fat accumulation in liver and insulin resistance. This is also influenced by sugar exposures, as are the gene expression changes related to these processes. But these effects are much more subtle than the changes in energy metabolism. So these findings show that, first of all, that the model is, at least to some degree, suitable to study sugar metabolism, but also that some of these rare sugars, not all of them, particular rare sugars also have quite some potential as healthier sugar replacers. If we now take a look at the potential to replace animals, I would like to take a look at the table below. And here you can see that human cells, so we were using cells from human origin, have a sugar metabolism that is at least sufficiently similar to human sugar metabolism. I would say more so than a rodent model, because rodents are much more likely to convert sugar into fat, which is problematic if you're testing non-alcoholic fatty liver disease development in humans. And if you would need a model for sugar digestion, you would already need to start using pigs. So, of course, the problem with such a monoculture model, one of the problems is that you only study one organ, and inter-organ interactions are an important part of physiology in vivo in any animal or human. And this is something that we can partly cover by those co-culture setups, because you can introduce different organs, not all of them, but at least the most important one. You can introduce those interactions. I would say for our type of research, in which you test many different sugars, you are screening the effects in the early stage of research, and then I would say such in vitro models are actually preferred, because they are more easy to do at high throughput and also at a lower cost. But you see that if you want to publish those results, some journals still desire verification of these results in animals for the later stages of research. But that doesn't mean that these models have little potential, because you can even, from this principle, you can even go a couple of steps further. If you would now use human organoid models which are like 3D organs outside of a body, and also incorporate the aspect of inter-organ interaction by using organ-on-a-chip approach, then you can have a really good model also in vitro that can really compete with those animal models. And these kind of developments that we are working on are relevant to finally get to such models. So to conclude, our model has been shown to be effective to study sugar digestion and metabolism. It has previously been shown, the HepG2 model has previously been shown to be effective for uptake of iron and polyphenol bioavailability as well, but now for a new purpose. We've also shown that that you can combine these different cells without any problems or harm to the cells. The model, especially the triple co-culture model, could be optimized further, for example, by measuring the nutrient flows and then obtain the ideal concentration that ends up in the third layer with skeletal muscles. And regarding animal research, this is ideal for the early stages of research and can also help to develop those more advanced models for later stages of research. So to conclude here, here's a list of publications that you can read if you're interested. That is the co-culture publication on the triple co-culture. We were the first to write an extensive review on the metabolic health impact of rare sugars. And we have a publication on rare sugar production and screening of the effects, together with our project partners, which I would like to acknowledge as well. So with many different project partners, we studied not only the health effects of those rare sugars, also impact, for example, on the intestinal and oral microbiota, how the taste of those sugars is perceived, and also a process to get those high sugars in higher quantities by converting just the conventional highly available sugars into those rare sugars. So I think now there will be room for you to ask some questions. If there is still something you like to know afterwards, you can also send me a mail or contact Charlotte Grootaert, one of my supervisors and the lab coordinator at NutriFoodChem.
Thank you very much, Amar, for your presentation. It's nice to see that your co-culture model has a lot of potential. I actually already have two questions after your presentation. First, I was wondering if you also have immune cells in your co-culture model, or if that's planned, if you know if the research project has that planned for the future.
So this model did not contain immune cells. Immune cells were one of the options that we had on the table when planning, along with also the fat cells, but in this case we chose for the liver and skeletal muscle. So it has not been incorporated in my experiments, but we, other people in our lab also do co-cultures with immune cells. So if people who are interested in that can still contact Charlotte Grootaert for information on that.
Yeah, okay, thank you. And I was also wondering, because your project in particular is about rare sugars, so I was wondering if the digestive model can also be used for the digestion of proteins.
Most likely yes. I know, for example, also other people in our lab are using similar models for protein digestion. I'm sure this model can be used, but I would say if you use it for a different purpose, you need some additional optimization for that purpose, but it will be useful.
Yeah, okay. Thank you very much, Amar. So, Maud?
In the chat, I don't see any other question at the moment, but maybe I can also ask one myself. So you also spoke about organ on chip, the possibility to improve the interaction between organs. But how would you do it if you had to create to set up one of this kind of model? Which organs would you choose to put it on? How would you proceed?
So in this case, I would think about, well, it all depends, of course, on the research that you're doing. I would think about similar organs. So we have now focused on intestine, liver, and also the skeletal muscle. So these are relevant, but you could also think in a diabetes context, indeed, on immune cells, but also a pancreatic model or fat tissues. So there are really many opportunities. Well, but to be clear, I have not been designing such a model yet. I was just discussing that following the same principle that could be an opportunity to really compete with these animal models.
Yeah, so connecting different type of tissues that have an impact on metabolism. Okay, so thank you for the reply. At the moment, I don't see any other questions.
So thank you again, Amar, and also Léa, for your nice presentations. If you still have any other questions left, I saw that Léa already put in the chat, you can contact her for more information. And I think Amar is also open to questions. And if you would like to know more about the other project of his former group, you can contact Charlotte Grootaert, I believe. If you want to be, if you would like to give some feedback on this webinar, there will be a survey if you close it. If you still need to have your certificate of attendance, please let us know. If you follow all sessions, it will count as half-day continuous education for the Brussels region. And finally, if you want to be updated about our activities, you can register for our newsletter on our website, www.replace.be. It's a hyphen between the re and the place. So don't hesitate to contact us for more information. And yeah, thank you for being here. Thank you for being very constructive. And of course, a very big thank you to the speakers.